All right, so experiment 27 is the IMViC series, a set of biochemical reactions. So, just a little background right here. So if you remember, we talked about coliforms, and they are a gram-negative or facultative anaerobic bacteria that produces lactose within 48 hours. And they're part of your normal flora in your gut, but can be pathogenic if they get out of areas that they are not supposed to be in. So like if you have, a coliform in your, I don't know, like heart or brain or something like that, it's probably bad. Coliform's kind of poster child is E. coli, and that's the major thing we test when we try to find about field contamination. So if you remember, we talked about the presence of coliforms when we did the most probable number technique test, right? So we look back at those at that experiment. It should be experiment 16, I believe. Yeah, experiment 16. So we did the MPN test, but this is more. So what this experiment kind of goes into is we know that there's coliforms in here, but we want to know which coliforms it is. So you can use IMViC to test which coliform contamination you have. All right. So it's 4 tests. It's called IMViC because it's Indole, Methyl Red, Vogue's Proskauer. I'm going to say Proskauer. It's probably not how you pronounce it, but Proskauer. So the I is whatever. I don't get why they didn't say IMVoc because it's Vogue's like MVO. That would make sense, but whatever. IMViC. So Vogue's Preserves is VI, and then Citrate is C. And so what it does is it uses these 4 tests right here, and you can learn a lot about the physiology of the microbes, right? So E. coli, something like this, is positive for indole, positive for methyl red, but negative for Vogue's Proskauer and citrate, right? And so all of these different microbes right here have different results. Well, I mean, you have some overlap, right? So like Proteus mirabilis and Citrobacter frundi is the same. So you have to do something else to differentiate between them. But a lot of these are different, right? So it's a way that you can like see like what you got where, okay? Or it provides an indication for that at least. All right, and so you're gonna want to keep track of stuff like that. So as we go through the video, you're gonna want to fill out this chart right here. So it keeps track of what the medium we use, so like what type of plate or agar or whatever it may be, what the substrate is for your reaction, what the enzyme that we're testing for is, what is the product of that enzymatic reaction, the reagents, any reagents or indicators that we need to use, what it looks like if you're negative, and what it looks like if you're positive, okay? So the first thing we're going to talk about is indole, all right? So indole is, starters, we had tryptophan, right? And we talked about, and tryptophan is another amino acid. So it has, carboxylic acid amino group, and it has that R group. The R group for tryptophan is this looking molecule right here. So it's got that, it's called a benzene ring right here, and then it's got this other aromatic structure. It's one of the aromatic amino acids, but it has this structure right here. All right, and when it's broken down, just like cysteine was, like it had that special enzyme, cysteine desulfidase. This one, the enzyme is tryptophanase, but when this is broken down, it produces this molecule right here plus pyruvate again, right? And so we can then use this pyruvate to use metabolic things. So it gives an indication of the metabolism. But this indole right here is produced from this reaction, and this is called indole. So what we do is we use a reagent called Kovacs reagent, and it reacts with indole to make a red color on top of the solution. So if you're positive like this, it's going to react with your indole and make a red color compared to this negative result right here, where you can still see the Kovacs reagent as that kind of yellow color right there. So if there was no tryptophanase in the enzyme, then there will be a clear layer on the top of the broth like this. But if there was tryptophanase, then it's going to be broken down into indole and make this red color. All right, so that's the indole test. The next one is the methyl red test. All right, so the methyl red test is when you use, so you're going to use tubes containing glucose. All right, so if you remember, glucose can be fermented. All right, it can go through both aerobic respiration, anaerobic respiration, and fermentation. But it can be fermented by bacteria. All right, and so the fermentation products, again, if you remember, they're acidic products. And if you have, so we talked about one type of fermentation, or we didn't really talk about it. You probably should have talked about it in class with either ethanol fermentation or lactic acid fermentation. But you make acidic products. If there are many different products produced from fermentation, so you're going to go a whole bunch of different types of fermentation, the solution can get quite acidic. It's called mixed acid fermentation. And so when you have so many acids, it can drop the pH pretty low. So what we do is we add a methyl red pH indicator. All right, and it's got a phosphate buffer in there as well. And if the acid is so strong, if this mixed acid fermentation is so strong, it can overcome this buffer. If you know what a buffer is, it's probably, but a buffer is something that's supposed to resist pH changes, but it only can go up to a certain point. So if it goes past that point and goes over it, it overcomes that buffer, then the solution starts becoming acidic, all right, and it can change the pH. And once the pH changes, then the indicator will change from yellow to red. So it's kind of changing to methyl red. Don't get this confused with phenol red. Phenol red is the opposite, in that phenol red is red at normal pH, and then once it becomes acidic, it turns yellow. This is yellow at normal pH, and then once it becomes acidic, it turns red. So you need to make sure they have those two distinct things right there. But that's the methyl red test. So it kind of tests for fermentation, in that you want to find the acidic products. Okay. The next one is the Voges whatever. All right. So this tests for a different type of fermentation. So some organisms produce much less acidic products when they go through fermentation. So something like acetylmethylcarbinol, which is another name for it, it's called acetoin. Not acetone. Don't get acetone confused with acetoin. Acetone is something completely different. This is acetoin. It's kind of a weird word. It's kind of a fun word to say. Acetoin. All right. So it can produce acetoin or butanediol. So this right here is acetoin. Right. It's got that double bond O with that OH right there. This is butanediol. Butaniol's got a couple different structures. You can have the OH in a whole bunch of different ways. But this is one of the butanediol. So they're kind of similar, right? This one just has a double bond right here. This one has a single bond with an OH. But it's just two different acids that can be produced here. So, but they're less acidic acids. So we test for acetylmethylcarbinol when we do the Voges test. So if you add alpha naphthenol and KOH to the broth, and then you expose it to oxygen, so you need oxygen, KOH, and alpha naphthenol, then you can produce this kind of red color from the from the reaction. All right, so you're positive if you have a red color, negative if you don't have that red color. So so what we do is we take the methyl red test and the Voges test, and we kind of it uses the same broth. Okay, so these are just a couple notes I get to make about this thing, is that we use this MRVP broth because all we need is fermentation to occur for both of these tests. We just need fermentation to occur, so we just need something that has glucose in it. All right, so we use the same broth that has glucose to test fermentation. When you're doing this experiment, all right, it's important to note this, so pay attention. All right, when you're doing this experiment, the manual says to pour half the broth into another tube. All right, so this is the diagram that you want to be looking at. So you want to do this correctly. Okay, so this is the new tube that you're going to be. So you're gonna put your microbes in the MRVP, and then you're gonna let them sit overnight, and you'll come back the next lab day. All right. And this is going to be your old tube that you used. Then they're going to give you a new tube with that next lab day, all right? You're going to pour half of this old tube into the new tube to where you have half of the tubes both filled, like this, all right? So your lab bench is going to be given, what, 1, 2, 3, 4 tubes, all right? Do not grab 2 of those tubes and do this, where you have 2 new tubes right here, and then you pour half of it into the old tube, half of it into the new tube, and your old tube's empty and your 2 new tubes. So don't do this, all right? You only want to grab 1 tube per old tube that you have. All right, and so you pour half of it, and then you do you do your methyl red test right here, and you do your Voges test right here. All right. And then just one more thing, when you're doing your Voges it says to shake constantly to get that red color. You don't really have to shake that much. if you just kind of let it expose it, you can kind of see the red color develop on the top of the thing, on the top of the like interface right there. But if you want to, you can shake to your heart desires, I guess. Okay. All right. So the last test is that citrate test. All right. And what this does is it uses the Simmons citrate auger. All right. And the Simmons citrate auger has sodium citrate, ammonium salts, and bromthymol blue. All right. So it's important to know that some coliforms do not have the ability to transport citrate and use that as a carbon source. And so they do not grow well on citrate media. If they do have that transport protein, then they can grow well on the media, and the products of citrate is a whole bunch of basic products. So that'll raise the pH. It'll make it more basic, right? Bromthymol blue is another pH indicator that is green at neutral pH, but then turns blue at a basic pH. So both the methyl red and the bromthymol blue, like methyl red, it turns red, and bromthymol blue, it turns blue. So in the IMViC series, it's the pH indicators are what color it turns if it reaches the appropriate thing. It's neutral at green and yellow, right? So again, you're just going to be growing microbes on this stuff. Okay, so the results. Yeah, so you're just going to be growing the microbes on this special auger, and then the results, it's positive if it turns blue, it's negative if it stays green. Okay, so this is kind of the filled out chart that we're going to do as a kind of like a takeaway type of slide. That the medium for the indole is tryptone broth. You're going to have the substrate being tryptophan, the enzyme is tryptophanase, and the product is indole, right? The reagent they use is the Kovac reagent, and it's colorless if it's negative, and it's red if it's positive. All right, with your MRVP broth, with your methyl red, you're gonna do MRVP broth. Substrate's glucose. Enzyme, there are many different enzymes that make mixed acids. Your reagent is methyl red. The appearance is yellow if it's negative, and positive if it's red. For your MRVP broth, you use glucose again, a whole bunch of different enzymes, but your product here is now acetylene. The reagent is the alpha-naphthol KOH, and then you also need it to be exposed to oxygen, right? You have a negative appearance if it's, your negative appearance is a colorless reaction, and then if it's red, then it's positive. So a lot of reds if it's positive, right? Your Simmons citrate, you use that test for the citrate, and the presence of a citrate transport enzyme, it's called citratase, I think. Your product is going to be an alkaline product. Your reagent's going to be bromthymol blue, and it's green if it's negative, blue if it's positive, right? So just a takeaway slide here is that so coliforms can be harmful if they're ingested, so we need to test for them. We're using an MPN test. Sometimes we need to differentiate between all the different coliforms, and you can use the IMViC series to differentiate here, and it shows you all the different physiologies that you can have. So depending on their reaction, depending on like Escherichia coli's reaction to all the tests, then you can say, oh, okay, there's this in this water, and we can treat for this. So it's really important that you kind of get all these reagents and all this stuff kind of fleshed out, and this chart should help you.